Good morning, everyone. I welcome you back for the next session. That is Ingress of the Next Generation Sequencing from Research to Diagnostic in the recent era through the eye of a biochemist. So, for today we have our speaker, Dr. Eli Mohapatra, Professor and Head of the Department of AIIMS, Raipur. And we have a chairperson, Dr. Sucheta Kumari N. She is Professor, Department of Biochemistry, HS Agri Medical College Academy, NITTE, Bangalore, Karnataka. So let me introduce the chairperson. And both of you, I welcome both of you. So Dr. Sucheta Kumari N. Madam has more than 30 years of teaching experience. Madam is highly acclaimed researcher for the past 2 decades in the subject of biochemistry, molecular genetics, cytogenetics, biodosimetry, lipidomics, and immunology. Madam has more than 400 research articles in national and international peer-reviewed index journals, and that close to 5,000 citations, and H index is around 40 plus. And coming to the research, Madam is a dynamic researcher, dynamic leader with extensive research expertise and experiences as principal investigator and co-investigator in managing several international and national funded projects like National Institute of Health funded from Wake Forest University, Johns Hopkins University, Penn State University, USA, and funded by Next Government Japan of University Mikazaki, Japan, together working around 2.4 crores from National ICMR, BRNS, Vision Group of Science and Technology, Karnataka, then CPCRI, INMS, and DRDO, together working around 2 crore. Madam has mentored around 26 PhD students. Coming to awards, FABMS Fellowship, Lifetime Achievement Award in the field of Radiation Biology, Women Researcher Award, Best Researcher Award by NITTE in 2018, Lifetime Achievement Award, Dr. P.P. Jacob Award for the Best Scientific Paper published in JIOS, author of Best Research Paper of 2020, NITTE, and many more awards. So Madam has guided 10 STS projects, and Madam is an executive committee member of Indian Association of Biomedical Scientists and International Society of Radiation Biology. It's not possible. Madam has so many achievements. It's not possible to share everything here. So I welcome you, ma'am. I welcome you and to judge this session, to chair this session. That will be taken by Dr. Eli Mohapatra in HOD of Biochemistry, AIIMS Raipur.
Thank you, madam. I'm very grateful to given the opportunity to chair the session, and I'm very grateful to the organized committee. I know how difficult to organize conference, and you have did it in a nice way. Very good organization and good speakers. Everything was very well managed. Thank you very much. And coming to our today's speaker, Dr. Eli Mohapatra, it's a great pleasure for me to introduce Madam. It's a great pleasure on my part. Dr. Eli Mohapatra had done her MBBS degree from MKCG Medical College, Brahmapur University in 1993 and completed her MD Biochemistry from SCP Medical College, Cuttack in 1998. In this young age, she joined AIIMS Raipur from its inception. It's a very good job. Now 2022 as an additional professor, head biochemistry, and pioneered the department and state-of-art laboratory of biochemistry department. Once again, a great achievement of Madam. Now she became Dean of Examination, AIIMS Raipur. She pioneered the sickle cell unit at AIIMS Raipur. Great achievement, Madam. She is the recipient prestigious Pittabas Jammu Memorial Award for medical paper in 24th ACBI Conference 1997 at Kolkata. And also she has taken the received the high-tech medical college certificate in her tenure from Biorad for internal and external quality control. Madam has teaching more than 20 years and she has become the chairperson of library committee, sickle cell committee, and she is the presiding officer of our internal complaint committee of AIIMS. And she is guided more than 15 MD students, and also she guided more than 20 MSc students, and she was a co-supervisor for PhD students. And also she is very good in working on drug designing for anti-cancer drug and anti-sickling drug. She has more than 30 papers published in her credit, and she has she was a collaborator of prestigious projects, ICMR, DST, as a co-investigator for more than 30 projects. Very interesting, Madam CV. She is also life member for many associations, AIIMS, ACBI namely, and she has become the reviewer of many international national journals, namely SDP, IJBB, and also she is the evaluator for project proposal of Chhattisgarh Council of Science and Technology, evaluator of thesis for MD Biochemistry, Utkal University, and she is the external member Board of Studies of AIIMS, Nagpur, and a member of Board of Studies under Utkal University. Madam is teaching for MBBS, BDS, MD, and also she is helping to creation of course studies, all these subjects. Madam, I welcome you. We are very happy to hear you, Madam. Thank you so much for my presence also here in front of you, all of you. Thank you. Welcome all madams. I am very grateful to our Dr. Manju madam who has given me the opportunity to chair this prestigious session. Thank you, madam. Over to you, madam.
Thank you, doctors. And it's a pleasure for me. It's a good, very lovely morning where we had two speakers and on different topics all around. And thank you, Sucheta madam, for those kind words. I'm grateful to you all. I'm entirely grateful to the organizing team for inviting me for such a conference, and which is being done very excellently. So now, like you can see in my slide, this is the place where I work. This is the college side of our institute in Raipur. This is about the hospital, which is a 900-bedded hospital having different blocks, and it's catering all the patients from Chhattisgarh as well as from the neighboring states like Orissa, Jharkhand, and Maharashtra, etc. So to start with my topic, I won't take much time. I'll start with my topic. And it's on the next generation sequencing. As we know, the technology for determining the sequence of DNA RNA is to study genetic variation associated with diseases or biological phenomena. And the commercial form of this NGS was introduced in the year 2005. Initially, it was called the massively parallel sequencing, as sequencing of many strands of DNA was being done simultaneously. Now, right now, during this pandemic, the NGS was there is increase in the centers of NGS over India. It has been found that there are available of around 24 in the different states, 24 states in India, 3 union territories, and 63 are operational sites. And out of which,
excuse me, madam.
Yes.
I think you have to go the slide show, and slides are not moving.
Slide show.
Eli madam can make that
yes,
slide
yes.
show.
Slide show. Can you see the slide now?
Yes,
NGS
no, it is not in slideshow form.
slideshow.
Yeah.
Now, is it a?
No, no, you have to say it again, I think.
No, she's not. Ma'am is not changing the slide.
Are you able to see?
It is there, but it is not in slideshow form.
Now it's moving. Now it's moving.
Is it okay now?
So-
No, actually, this is Windows 7, so you can't do the slideshow.
Okay. Here, resume the slide.
Yeah.
Is it? Is it working now?
No, ma'am.
Click on share screen, ma'am,
Yeah.
and it's share your slideshow screen. No, not PPT. Share your slideshow screen on Zoom.
Okay. And now see your application. Is it visible now?
Yes,
Yes,
madam.
it is visible, ma'am, but the slides are not moving actually.
Previous slide. Previously. Previous. Is it moving now?
Okay.
Is it
Yes, ma'am.
No,
moving?
ma'am. No, ma'am.
No. Now is it visible? And can you say, like, is it changing?
Visible, but
Is it in the initial slide? Is it moving?
Initial slide.
Yes, it is moving, ma'am, now.
Now is it moving?
Mhm.
Okay.
Yes.
So like I was talking about the NGS technology and now it's being known, initially it was known as massively parallel sequencing, and it was doing a number of strands were being taken simultaneously. Then now, like, during this pandemic, we had so many centers opened up in India. Almost 63 operational sites where NGS being done. And it includes in almost 24 states and 3 union territories. And there is also upcoming centers of NGS in 20 or more than 20 sites in India. So this was the blessing in disguise during the pandemic. So normally we know that during in any DNA sequencing, one has to determine the order of nucleotides in a DNA, which includes the methods mainly basically it is dependent on determining the order of the 4 bases, the main adenine, guanine, cytosine, and thymine. So the DNA sequencing, initially it was in 1970s, before this structure of the DNA was being determined in 1953 by Watson and Crick. And thereafter in 1977.
Ellie, ma'am, sorry to interrupt. This slide is not moving. You have to click there, I think.
I'm clicking.
This is second slide only. So third slide.
I didn't hear.
Is it clicking now?
No, ma'am. Only we can see the second slide.
Ma'am,
No, it is coming, third slide now.
Click. You click on the slide, not on the board. Side panel.
Click on the slide, ma'am.
Okay.
Yeah, now it's. No,
Yeah,
yes.
it is coming.
Now
ma'am.
is it
Yes,
visible?
yes.
Okay.
Now it is correct.
It is coming, ma'am. now.
Yes.
Yes, fine.
So, this is about the timeline of the DNA sequencing, which like the structure of the DNA, we know that it was being started by Watson and Crick, and later on 1977, Sanger method was developed with the, they used DNA by the chain terminating method, and thereafter a number of researchers, they went for different, defining different ways out, and in 1986, one can see that we got the automated DNA sequencing system. So the sequencing methods that mainly consist of the classical, as we all know, the Maxson-Gilbert's chemical degradation method and the Sanders method. The second is the second generation sequencing, where there is next generation, I meant to say, where there is second generation sequencing, third generation, and the fourth generation. And this fourth generation is now with a single molecule sequencing in the third, it was adapted in the third generation as well as in the fourth generation. That is how it differs from the second generation. And this started during 2010. So like mainly the approach of NGS technology is through short read sequencing and long read sequencing. As we know that they have got a lot of advantages as well as limitation. The short read sequencing has got higher sequencing fidelity. It's cheap and can sequence a fragment, whereas long can take the entire. So if you have missed out any of the repeat sequences in a short one, you can have it done in the long read sequencing. That is how it works. And you can identify the structural rearrangement in the homologous regions. So, and but the disadvantage is found to be that there is low per read accuracy in case of long read sequencing. Now, the second generation sequencing techniques, they include 454 sequencing or pyrosequencing. It was named after the project 554, therefore it's been called. Then Illumina's high-sec and MI-sec sequencing, then Sol-IT sequencing, Polony sequencing, and the massive parallel signature sequencing. Now, the two second generation technology, the advantage was it was fast, it was cost effective and very sensitive. But there was some drawbacks, which included the poor interpretation of the homopolymers and the incorporation of the DNTVs were sometimes incorrect, which resulted in wrong sequence or resulting in a sequencing error. The major drawback was the need of PCR amplification, which was needed prior to the sequencing and which resulted in PCR biases. So to overcome this, the third and the fourth NGS technology came up. Now, coming to the 454 sequencing or the pyrosequencing, it was being reported in 1993, where a mixture of DNA polymerases, ATP sulfurylase, Firefly, luciferase, and NNTPs is added to the single-stranded DNA to be sequenced, and then the light emitted by it is being measured, and the intensity of the, depending on the intensity of the light, one can incorporate the different, find out the nucleotide present and find out the complementary nucleotide that is present on the template strand. The Illumina Solex high sequence and MI sequencing, it claims to be the most successful sequencing system, and claims to be having 70% dominance of the market at present. And the sequencing by this synthesis, it uses removable fluorescently labeled chain terminating the nucleotides, which are able to produce larger cost-effective output. And the clonally enriched template DNA for sequencing is being created here by the PCR bridge technique. The amplification that is being used is PCR via bridge. And the output of this sequencing is found to be quite higher. It's found to be 600 GB. The SOLI sequencing, which was given by the Applied Biosystem, it based on sequencing by ligation. It uses a pool of all possible oligonucleotides of a particular length, and then the oligonucleotides are being annealed, ligated, and depending on the sequences, and then the signal information is being found out. The ligation, as we know, is being done by the DNA ligases, and before sequencing, the DNA is amplified by immersion PCR. The Polony sequencing, which was developed by George M. Church Laboratory in Harvard, was the first high throughput sequencing system, and it used the full genomic sequencing of E. coli in 2005. And now for this, they had an amalgamation of in vitro pair tag library with immersion PCR, an automated microscope, and a ligation-based sequencing chemistry to sequence the E. coli genome. And it was found that the accuracy was found to be 99.9%. So it was very accurate and cost effective. It was 1/9 of the, approximately 1/9 of that cost of Sanger sequencing. The last in the second generation is the massively parallel signature sequencing, which is a bead-based method, and that applied to a complex approach of adapter ligation followed by decoding, adapter decoding, and reading of the sequence in an increment of 4. For this, as usual, one has to go for the extraction of the mRNA from the tissue, convert it to the cDNA, and then the following steps are being followed. It undergoes PCR amplification, load the beads by hybridization, and at last, it results in match of genomic sequences to determine the transcript for each signature. After this comes our third generation sequencing technique, which includes a single molecule real-time sequencing and the heliscope sequencing. The introduction of 3G sequencing, it circumvents the need for PCR. Here, sequencing a single molecule of DNA without any amplification, one can go for it. So thereby we can overcome that PCR biases. Here the sequence information is obtained with the use of DNA polymerases and by monitoring the incorporation of the fluorescently labeled nucleotides to the DNA strands with a single base resolution. Now the SMERT, or the single molecule real-time sequencing, is being executed in a SMERT cells, and each SMERT cell accommodates 150,000 ultra microvels. Each well having a scale of zeptoliter, where one molecule of DNA polymerase is immobilized at the bottom of each well with the help of the bioinstructor within system and nanostructure known as the zero mode waveguides. And then next, what happens is with the help of the CCD cameras, one can scan the zero mode waveguides in a series and distinguish the pulses, and thereby can make out which are the different bases being present. So this is in detail about the SMERT sequencing, where the microvel or one molecule DNA polymerase is immobilized here, and then with the help of the bioinstructor within system, then once the template assist the single strand DNA binds and with the immobilized DNA polymerase. It is fluorescently labeled with DT and TPs and are incorporated, and then nucleotides are added to the growing strand. So after polymerization, then one can see, and then you get the primary analysis data, and then from the primary analysis that data secondary stream directly into the secondary analysis software called SMERT analysis, and also known as the process of sequencing in real time. Now, as all the core nucleotides are added simultaneously, the measure is real time, and the speed is quite rapid here. The heliscope sequencing was being given by the Heliscope Biosciences. Here, the sequencing, sequences millions of billions of molecules in parallel, and the DNA is cleaved and tailed, and with a poly tail fragment, and hybridized directly to the oligo TT50 bound on the surface of the disposable glass flow cells. The incorporation of the fluorescent nucleotide with the terminating nucleotide discontinues the cycle until the last one nucleotide of each DNA sequence has been captured. And then the process is repeated until the fragment have been completely sequenced. And this technology is a combination of sequencing by hybridization and sequencing by synthesis using the DNA polymerases. The fourth generation sequencing technique, which includes the DNA nanopore sequencing and the nanopore DNA sequencing. Now here in the fourth generation sequencing, you'll see that the complete genomic has been made out of this fourth generation sequencing. It's high throughput sequencing, and it can determine the whole genome of the organism, as I told. And the method employed here is rolling circle replication to amplify small fragments of the genomic DNA into DNA nanoballs. The unchanged sequencing by ligation, and then applied to determine the nucleotide sequences. The second one is the nanopore DNA sequencing, where there is a passage of DNA through the nanopore, which produces current, and which is being depends on the size and length of the DNA. And the images you get in the computer depending on the type of current and the height, and you can determine the bases that are being present. The 4G system currently offers the fastest whole genome sequencing scan. It is still quite expensive, and it's but it has got number of error. It's prone to errors as compared to 2G technique, and is relatively new. Currently, less extensive data is available for this technique. This is a basic flow of any NGS. One has to go for first construction of library. Then there is clonal amplification, sequence of the library, and followed by lastly the analyze the data. Before one goes for this, one has to go for the pre-processing, that the nucleic acid, either the DNA or the RNA are being extracted from the selected sample. Then the extracted sample undergoes the quality control check by any of the standard methods. And then in case of RNA, one has to go for transcribe it into cDNA. Then one goes for the library preparation by random fragmentation of the cDNA or DNA by enzymatic or sonation. These fragments are then end repaired and ligated with the smaller genetic DNA fragments called adapters. And the adapters have defined length, and they are oligomere. And these adapter, along with the DNA fragments, they give rise to formation of the sequencing library. Then there is a size selection and the library enrichment or amplification using the PCR, that is the emulsion PCR. And this amplification is followed by cleanup step, and then QC checkups are being done. We're using the qPCR to confirm the quality and the quantity of the DNA. And the sequencing is dependent on the selected platform and chemistry for clonal amplification of the library fragments and can be done prior to the sequencer loading, either in the immersion PCR or via the bridge PCR. Depending on the platform, it has been accordingly being told. The data, the last step is the data analysis, and that is being done to analyze the different data that is being got at the end of the stage. It varies depending on the type of work that the researchers is doing, and keeping what he expects, what is there, the question in his mind, he has to take up the data, analyze the data accordingly. This is how the NGS workflow, as I already mentioned, the create a fragment, and then there is a added to a specific adapter, the adapter binding with the DNA, which is followed by bridge amplification to form a cluster, and then there is a primer binding and sequencing. The bridge PCR, here you can see very well that how there is formation of a bridge kind of thing between the, so one end is attached to the DNA fragments are planked with the adapter, and a solid surface of this which is coated with the primer complementary to the two adapter sequencing, and following isothermal amplification one gets the complementary strands here. This is like I had another image where it shows the Illumina showing the bridge PCR in details. I think as I'm running out of time, I'll go past. Then next comes our cluster amplification, which is being done by immersion PCR. It involves the fragments with adapter, the library, and the PCR is amplified within a water droplet here in oil. The PCR primer is attached to the surface of the bead, and then the DNA molecule are synthesized on the bead in the water droplet. Each beads bear here the clonal DNA originated from a single fragment beads, and they are deposited in the wells sequencing chips. So each well has got one bead. So there are various sequencing technique, which includes this cyclic reversible termination, where the sequencing by cycle CRT is being done with a reversible terminator in a cyclic method, and usually after the incorporation, usually you read about 100 to 150 BP reads in this technique. Another sequencing technique includes your sequencing by ligation, uses the enzyme DNA ligase to identify the nucleotide present at a given position in a DNA sequence. So there is also pyrosequencing. Pyrosequencing is a non-electrophoretic bioluminescence method which measures the release of inorganic pyrophosphate, which is proportional to the visible light that is being converted during the conversion of this enzymatic reaction. And the nucleotide incorporation generates light, and which is being got in the form of a peak in the pyogram, and it is being incorporated. Then comes again the sequencing imaging technology that includes the SMRT, about which I had told initially in detail. It is a single molecule DNA sequencing method, and a single DNA polymerase enzyme is affixed at the bottom of the zero mode wave guide, and with a single molecule of DNA as a template DNA polymerase. So coming to the application of the NGS, it is being used mainly, like we use it in a number of ways. It can be used in cancer research of different organisms in order to find out the types of different diseases which are unknown to us, whose etiology is still not clear. Now, in case of cancer, you'll find that the benefit of cancer of the whole genome sequencing can be done. And the benefit is that here you can, a cancer genome can contain unpredictable number of points of mutation and fusion. And many a times, if you go for exomic sequencing, these points may be remain undetected. And so if you go for a whole genomic sequencing, it is a novel method and it will help in finding out the mutation in the entire genome you can study. And so that it offers a very comprehensive approach for variant identification. Whereas in case of exome sequencing, one may miss certain variants and which remains outside the coding regions. So the whole genome sequencing provides a base pair resolution of the entire cancer genome in a single run. And this method offers a comprehensive view and of any kind of unique mutation or genomic alteration which is found to be seen in the cancer tissue, including those contributing to the surrounding normal tissue and the tumor clonality. Cancer's exomic sequencing, here you identify in the coding mutation region or in the cancer. So the sequencing of the cancer exomic provides useful information about the coding mutation that contribute for tumor progression. And because of the exome represent only 2% of the genome, the cancer genome sequencing using NGS technology is a cost-effective alternative in contrast to whole genome sequencing. The advantage of cancer exome sequencing is that it allows cancer researchers to assess not only the coding regions, but frequently, which contains frequently the mutation, but also how the tumor progression is also occurring. Depending on the experimental needs, the content of the cancer exome sequencing can also be expanded to the untranslated regions and the microRNA binding sites. And because it offers an accessible combination of turnaround time and prices, the exome sequencing is the best, and is being considered as the best method of choice for the tumor and the normal comparison. Then, as I told you, it can be used for microbiology research, from the environment and metagenomics study to infectious disease, its surveillance, sequencing, and in order to get insight into the genetic study in order to know new bacteria or viruses. And also it helps in characterizing the unculturable organisms. It discovers entirely new viruses, as it has been done recently, and develop new strategies to control the outbreaks. And one can also monitor the host pathogen interaction during this time. So the third is the important use is in case of complex diseases. Many of the complex diseases, as you have seen, like in case of autoimmune diseases, rheumatic diseases, atherosclerosis, neurological diseases, and psychiatric diseases, one is not able to understand what is the cause or where there is dearrangement, so that the treatment protocol can be given accordingly. So on a molecular level, it has to be characterized, and more than 90% of the diseases are associated with variants that are located in the non-coding regions of the genome. 5% of the complex diseases are called by monogenic inheritance, while mainly vast where majority is polygenic in nature. That is all these neurological diseases, atherosclerosis, psychiatric disorder, and all. So the application is high throughput determination of RNA structure, and by sequencing technology, which enables the RNA to determine at remarkable unparalleled scale. Then towards the genomic information in real time for all global pathogens and surveillance system. Now, recently in the 2020 pandemic, NGS were the pivotal in characterizing the SARS-CoV-2 genome and constantly monitoring the COVID-19 pandemic patient. It also helps in enhancing the accuracy of the NGS by detecting rare and subclonal mutation, then harnessing the ancient genome to study the history of human adaptation. One can go for sequencing and you get a better understanding by this. The other application of NGS includes the rapid sequencing of whole genome, deeply sequencer target regions, utilize RNA sequencing to discover novel RNA variants and analyze the epigenetic factors which are responsible giving rise to cancer and other diseases. It can be used for study of rare somatic variants and tumor subclonals, and the study the human microbiome and the novel pathogens. So accessible of whole genome sequencing is used by using the capillary electrophoresis. It was found that the Sanger's method, the costing was nearly $3 billion, and which is whereas by NGS it was this costing has been reduced. It has cut through the costing. The NGS, in contrast, had made a large-scale whole genome sequencing accessible and practical. And now, like we all know, NGS-based RNA sequencing is a powerful method that enables researchers to break through the inefficiency and expenses of legacy technologies like microarray and the microarray genes expression measurement in limited, which is limited by noise at the low end and signals saturation at a high end. And as contrast to it, the next NGS is found to be discrete digital sequencing reads count and its broader dynamic range. It has a broader dynamic range. It's tunable, and it has been found that it's found to be quite efficient, cost effectively, and focusing on the power of NGS. It is highly scalable, and then it can choose whether one has to go for a shallow scan or a deeper scan in order to get the variant regions of a given region. But the bottleneck of this NGS sequencing is the complexity of the sample processing for NGS. And because of managing a high load of data, analyzing and storing the data sets. It requires a lot of amount of computational resources for the assembly, annotation, and analysis of the sequencing data, which is one of the biggest challenge. And then the vast amount of data that is being generated through NGS has to be analyzed, which is another critical challenge. So more strategies are being suggested with the aim to increase the efficiency and to reduce sequencing errors and maximize the reproducibility and ensure a correct data management. So as even told by the Dr. Vijay Shugrumaran, like every technology has got its advantage and disadvantages, but it is providing some sort of benefit to us, though according to the cost effectiveness, then getting the answer of any of the disease or research within a short period of time. But there are presence of certain limitation also, which have to be overcome gradually in the time to come. And with this, I'd like to end my presentation here. And thank you. I thank again the organizing committee for inviting me for such a wonderful and interesting, knowledgeable this conference. Thank you.
Thank you, madam. Thank you. It is a very eye-opening session of NGS, because we heard NGS. We actually, from my lab, we are outsourcing the samples. We don't have the setup. Do you have the setup, madam, in your hospital?
Yeah, we have recently during this COVID time, one of the NGOs was being, a multiplex NGOs has been there in our department.
Yes. How is the maintenance of the setup?
Maintenance, yeah, it's cost effective in that case. The maintenance is very important factor, and other than that is the consumables, which are like, if you don't get a large amount of tests to be done, the use of consumables and the price that the consumables are quite high. So like making it a little bit costlier at this time. Hopeful that gradually there will be lowering of the costing with more use of consumables and all.
Yes, madam. Actually, you are given a very good chronological development of NGS, and also I know we have a lot of setup in our lab. Major problem is site preparation, maintenance, and AIIMS, and technical support, troubleshooting, so many things. In addition, you are maintained NGS very good. I think we may have to go for 1 or 2 days workshop on this particular NGS part. And in the point of time, you have explained what all the things to learn for a young generation. I think it is we can take it as a one subject, one degree on NGS. That much part we can study under this ending. It's a very good. I wanted. There are some questions, madam, where I'll ask them to
Yes, ma'am.
connect to them. Please go through the questions, madam. Hello.
Any questions? There is no question in the chat box. Question
Okay.
answer, sir.
Uh,
So anyone can ask me, sir, they can ask.
May I ask?
Some questions they asked. One is, what are the major challenges we face during microarray sequencing? And one more question they asked, what will be your advice for startup service for INI randoming free service? Do you offer any hands-on training? All that, there are some people are asked questions.
We right now, as we were like during this pandemic, we were very much busy with this. We couldn't start with the, but yeah, of course, we had all hands-on training for all the technical staff of all the whole of the Chhattisgarh state. And yeah, we can have it after now that the COVID has come down a lot. The only few cases are erupting. So when we can have, and last year we had a MBBS conference, but we couldn't have the hands-on training on those things during because the pandemic was still going on. So now we can think of having such kind of workshop where we can give hands-on training on NGS also, so that the future generation will be greatly benefited.
Thank you, madam. Thank you for wonderful talk. And I thank you and Dr. Manju, madam, and whole team for giving me an opportunity. It's a great pleasure. Thank you. Thank you, one and all.
Thank you so much, Ellie ma'am. I have a question actually. Do you use this for diagnosis? Your setup?
Right now, they are actually, when the different variant came of this COVID-19, during the second wave, there we started it on the diagnostic purpose, but now like it is hardly being used, as this number of tests has reduced, this number of samples that we are getting, because due to the reduction in the number of COVID cases. So hopeful, and now the swine flu thing has come. So now the team has started with this also. So hopeful, like
So thank you so much, ma'am, for this wonderful informative talk about the advantages, disadvantages of different NGS techniques and its applications also in cancer research, microbiology, and complex diseases. Thank you so much, ma'am.
I thank you all for inviting me.
Thank you, Sucheta, ma'am. So now it's time to honor with a memento virtually and certificate. I would like to honor Dr. Eli Mohapatra, ma'am, for this nice talk. I'd also like to honor our chairperson, Madam Sucheta, madam, for this chairing this session. Thank you so much, ma'am. Thank you all. Now we'll move to the next session. So thank you all.